Thank you so much for the invitation, and thank you so much for putting together this really nice session. So I'm going to tell you about some of our recent work in interpreting deep neural network models for understanding how rules of immune cell differentiation, which I'll talk about, is encoded in our genomes. So motivation goes back to this fundamental question in biology, which all of you are familiar with, which is how is it that the same genome in an individual can give rise to so many different cell types and tissue types? The immune system is a really nice system to study this because for years now, or for decades, immunologists have agreed on at least 100 distinct, immunologically distinct cell types. And I'm showing you a picture of those on the right there, some of the immune cell types that you might have heard of. So of course, we also know that a big part of the explanation is differential usage of regulatory regions across the same genome. So here's an example of 3 immune cell types: NK, CD4s, and B cells. And in this cartoon, the idea is that each of these cell types might have their own distinct set of regulatory regions that are used to regulate downstream gene expression, and that gives rise to some of their differences. And more formally, the hypothesis is that within these regulatory regions, if you were to analyze the sequences, we would find that there is sequence composition differences that allow for coding or encoding of different combinations of regulatory factor motifs, in particular transcription factor binding motifs that might be distinct for each cell type or cell states. So the work that the research that us and many others have been interested in for years is how do we design models that can analyze regulatory regions or sequences, genomic sequences in regulatory regions to decipher the sequence syntax and rules and grammar that specifies cell type specificity. So looking at regulatory sequences with kind of machine learning models, can we predict, first of all, where is that regulatory element? What cell type is that regulatory element active or required for? And what are the sequence logic and rules encoded in that sequence that allows us to do that? And I've listed at the bottom a lot of really exciting new work that use this framework of sequence to function models that I'm going to tell you about with convolutional neural network to try to answer some of these questions. So in particular, in this talk, I'm going to tell you about work from 2 students in my group. I'll first talk about our AI tag project done by Sasha Mostafavi, where she developed a robust interpretable approach for figuring out how to understand regulatory principles learned from sequence information encoded in the regulatory genome across immune cell types. And in the second project, I'll tell you about Shiming's work and actually try to improve our ability to derive causal insight from these sequence to function or sequence to activity type models. So let me first tell you about the data, and then we'll get to the models. So the data for much of our work comes from a collaboration that we have with the Immunological Genome or MGen consortium. And their latest data generation effort, what MGen did was for 90 distinct immune cell types that were isolated in adult mice, they generated ATAC-seq data to define regulatory elements and regulatory regions across all of these cell types. So what I'm showing you here is, and let's see if you see my mouse, yeah. So what I'm showing you here is a dendrogram of immune cell differentiation tree. So on top here, you have stem cells, and the leaves are terminally differentiated cell types. And what this illustrates is that there's a large number of functionally distinct immune cell types that are captured here, and they belong to broader lineages. But within each lineage, we have lots of very granular defined subtypes. So here's another way to look at the data. We have ATAC-seq data. So what that means is that for each of these cell types, we can define regions that are accessible. And I've shown you by colors here. And for each accessible region, these are isolated cell types. So we have some reads that fall in those accessible region, and that can serve as some sort of quantitative signal for kind of regulatory activity or regulatory potential for each of these regions. And altogether, this data set gave us about 500,000 regions or ATAC peaks that are accessible in at least one of the 90 cell types that were profiled. So using these data as input, we created a model that we call AI TAC. And what this model does is takes as input regulatory sequences underlying each peak. So these are each about 200 to 250 base pair long. And on the basis of sequence information, predicts accessibility simultaneously across the 90 different immune cell types. So this is a multitask learning model that using the same input tries to simultaneously predict accessibility across all these cell types. And I should mention that it's a convolutional neural network model, which is a standard model nowadays for modeling kind of sequence information. So at a high level, how this model is going to learn is that different hidden layers in the neural network model in this setting is going to learn different abstract feature abstractions with increased abstraction. So for example, nodes in the first layer are going to be recognizing and learning short sequence motifs that might resemble transcription factor binding sites. And these motifs are then combined in the later layers to make the final predictions. So the idea is that if we can train this model, can we then use it to understand sequence motifs and their combinations that are important for making prediction at granular cell type levels? So I'll illustrate two aspects of the result. So first is that, well, can this even work? Can we make predictions from sequence information alone? And this summarizes the cross-validation result. So the idea is that, or the summary is that for about 60% of attack regions, on the basis of sequence information alone, we can predict accessibility across these 90 distinct immune cell types. So that's a lot of information that's contained in the sequence itself. And here is an example of a very granular and nice prediction at the right of the histogram there. So what I'm showing you here is the accessibility profile of one peak or ATAC peak across the 90 distinct cell types. And on the bottom, you have the predicted version of that bar chart. So the point is to make that these particular attack test attack peak has very variable accessibility across the cell types, and you can achieve the same level or make the same kind of predictions by the model. So now that in our paper that's published now, we kind of validated or tested generalizability of the model on many different out-of-sample data sets, including a cross-species example. And now that we were convinced that we can actually make prediction from sequence information alone at this very granular cell type specificity, we wanted to figure out how do we interpret the model to learn what actually has been learned. And if you actually follow this literature, there's a huge emerging field of explainable AI for figuring out how to dissect complex models like neural network to understand what they've learned. And we actually have a paper that's hopefully coming out soon on just a review paper on summarizing various approaches that you can take to this problem. But in this particular paper, we showed that a simple node-based strategy actually works pretty well as long as you account for uncertainty in the model parameters that have been learned. So our step consists of our approach consists of three steps. The first step, we're going to, for each node in the first layer, we can do it with nodes in the other layers, but I'll just talk about the first layer. We can identify specific sequence motifs that are being recognized by just looking at which subsequences are activating each node and build a position weight matrix from those. The second step, what we're going to do is we're going to ablate each node and measure how much did the prediction change with when the node was in place versus when we ablated the node. And that gives us a measure of the importance of that motif to the model's prediction. And third step, which wasn't actually done before, but is very important, is how do we account for uncertainty in the parameters? And for this, we took a simple strategy. So what we did was we trained 10 separate neural network models on the same data. So the training data is the same. The only thing that changes is the initialization, which also leads to differences in the parameters and the model optima. So if you repeat this 10 times, then for each motif that we've identified, we can say how often do we find the same motif in separately trained models. And so we saw something interesting here. So the plot here, each dot is one motif or one PWM learned by the first layer. And we have 300 nodes or correspond to 300 motifs just by the architecture of the model. And on the x-axis, we have the information content. So how different is that motif from just random sequence? And on the y-axis, how influential that motif is to the model's prediction. And these are color-coded by how reproducible they are across the 10 separately trained models. So what we showed that, and this is a general property, it applies to this setting in our data set, but it's more general than that, if you use this kind of node-based interpretation technique, what happens is that there is also lots of noise that are learned by the model. So 1/3 of the motifs that are learned are reproducible across many different trained models, but many of them are not. And if you actually take this into account, it turns out that different model interpretation, we can close the gap between different model interpretation strategies. And that's kind of what I'm showing you here, which I won't go through on the right. So what do we actually find from a biological standpoint? So we can go and now match these motifs that we found with known transcription factor binding databases like SysBP and JASPAR. And if we do this, once we take into account only motifs that are reproducible, we actually see the vast majority of the motifs that the model find correspond to known transcription factor binding sites that are known to be important immune regulators. You can see some of their names on this plot here. There are some that are that have not been annotated, but vast majority have been annotated, which so this is all known. So the model is rediscovering a lot of the transcription factor motifs that were well known. But what is really interesting is that the predictions are being made based on combinations of these motifs, and not any individual motif itself is not that predictive. So how many combinations there are, and maybe this is the last slide that I'll leave you with on the first part of the talk. So we can kind of quantify that for any given sequence, any given ATAC peak where a model makes a prediction, how many motifs do we need to make that prediction? And this is the histogram of that. Basically, on average, for any given ATAC peak, there is 2 or 3 motifs that are needed to make prediction across immune cell types. But there are many that need far more than that. And you can see like a wide tail of the histogram here. Okay, so the second part, way more briefly, I'll tell you about Shiming's project on how to improve our causal insights that we can derive from these kind of sequence to activity models, as we call them. So one really important utility of these kind of sequence-based model is that we can use them as a sort of an in silico mutagenesis assay. The idea is that once you train a model, if it operates on a sequence, what we can do is feed the model the reference sequence, something like we have here, and then we can inject or we can mutate any base that we want and ask the model, how does the prediction change once we have a variation, which a model might have never seen before. And the idea is that if you apply this kind of mutagenesis and silico mutagenesis systematically, we can create these ISM maps, as they're called, where for every single nucleotide, we can say what's the effect of that mutation on the model's prediction, and in this case, model's prediction about accessibility and for a particular cell type. So we can have very fine resolution of predictions about how mutations at any combinations of loci can affect accessibility. So this is a very powerful utility of these sequence-based models, but it's been really hard to benchmark these for a main reason, which is lack of really nice data that allows you to benchmark these. So we can make all sorts of predictions, but in order to figure out how accurately they're making these predictions, we need perturbation data that is done in a matched cell type and matched system where the data for training the model came from for it to make sense. In our context, it turns out that we can actually do this because there are multiple strains of mice where the same type of data can be generated. So the model that I showed you initially was trained on data from B6 mice strain. And this is the predictions that we can make for one cell type in terms of ATAC accessibility for B6. So this is in particular the CD8 cells. You can see the model's predictions are reasonable. They're just based on sequence information. So for that, given that, these are pretty statistically significant. But now use the same model and apply it to a different strain of mice, in particular in this here, the Cas strain. and castrain and B6 have about 20 million variants between them. And you can see that there is kind of a big drop in the prediction ability. So the model can probably make sense of some of the variations, but not the majority of them. So this gave Shenming the idea that why don't we use natural genetic variation as a sort of systematic perturbation data to increase our ability to make predictions about genetic variation from these kind of models. So with this, to implement this idea, we worked with Christophe Benoit's lab and a great PhD student there, Kat. And what they did was generate F1 hybrid B6 and Cas mice. And the special thing about these mice is that they are basically heterozygous at almost every loci across the genome, because there's a lot of differences between the B6 strain and the Cas strain. So we can, with this data, we can also generate ATAC-seq data. Now, for any given loci, for any given peak in the ATAC-seq data, we almost always have two alleles that we can look at, the paternal and the maternal allele. And those are going to have different genetic variations in them, and we can try to correlate that with accessibility differences between the two strains. So in order to take advantage of this data, what Shenming developed was a model that we call deep allele. So what it does is that for any given loci, it takes both alleles as input, just the sequences across both alleles, and it's a multi-head model. As output, it predicts the counts from each allele as well as the ratio of the counts between the two alleles, as a way to enforce the model to learn really which genetic variation or differences actually are responsible for the differences in the counts that we might see in the two strains. So just very briefly, I'll illustrate two aspects of this result. So first is that by using this model where we actually using multiple genomes as opposed to just one, we can improve our ability to predict the effect of in silico mutations. And we can show this by using basically leaving out some of the accessibility data for some of the peaks and checking to see the model, can the model make accurate predictions about genetic variation for the peaks that it hasn't seen before. And this is, the blue curve is the new model, and the orange one is our previous model that actually was not trained on multiple genomes. And the kind of the last aspect of the result that I'll illustrate this plot here, now that we have this multi-head model, we can distinguish two types of motifs that the model finds. So each dot here corresponds to one motif, which is one node in the first layer that I mentioned previously. And we can look at the influence of that motif in terms of the model needing that motif to predict counts versus the influence in predicting the ratio between the two alleles, or the allelic ratio. And the motifs that are important for predicting allelic ratio are the ones that actually have some causal evidence according to the model, because if there's a mutation in those allele, you're going to see different in accessibility count between the two strains. That's all I had to tell you today. I'll leave you with that. And for acknowledgement, Sasha and Gurman and Shenming that I mentioned, as well as our collaborators, Christophe Benoit, Ricardo, and Kat from his lab. Thank you so much, and happy to take questions.
Thank you, Sara, for the great talk. If there are questions, please come to the microphone to ask questions. I may start with one. Actually, I have two questions. I'll start with one for now. Do you think your approach to analysis of the F1 hybrid data is generalizable to the diverse, let's say, genotypes of human population?
Exactly. That's exactly where we're going with that. So basically, you can think of each person, so the question was that, is this approach generalizable to looking at genetic variation across actual people? And that's exactly where we're going with this. So you can think of each individual as like an F1 hybrid in a way, as long as we can phase their allele. I think computation cost is like the major bottleneck, but we're thinking of strategies of addressing that.
Hi, Sarah. So I was wondering, you've trained, in both of these projects, you trained multitask models. What we found in some of our work in the past is that when you try to train a multitask model and then do feature attributions for each of the individual tasks, that you see leakage across the tasks. Like if you have a protein that binds in one cell type but is not even expressed in another, the cell type where it's not even expressed, you'll still pick up the motif because it was trained in the same model. Do you experience any of that with either of your models?
I think it depends a lot on how you do the feature attribution. So, I mean, I do see some of that when we try to fine tune models from one task to another. Then there's some sort of leakage. But I think a lot of it depends on, are you attributing to individual outputs and you still see the
Yeah.
leakage? Yeah, we can talk about that. I don't know. Yeah,
Okay,
that's a
thank
good
you.
question.
Hi, Sarah. Great talk. I might have missed it. I might have phased out at some point. But do you know that you need to phase the alleles? Like, what if you just input genotype rather than like the haplotypes? I mean, you can imagine you might have to phase the alleles.
Yeah,
but
actually,
have you
that's
tried
yeah.
not phasing?
That's a great question.
'Cause-
We have tried phasing, and that's a big pain. So I think if we can get away with it, it would be really nice. And that's, yeah, I think that would be nice to try to see if it actually makes any difference.
Okay, thanks.
One question from the audience, from the remote audience. How does deep allele compare with traditional allele segregation method like SNP split?
Okay, that's a great question. I don't know, what was it called? SNP split?
SNP split.
Okay, I know a two-method Maggie, and there's another one called we call it MinDiff. So yeah, we are benchmarking against those methods right now. And what I can tell you is like basically the motifs that we are discovering are consistent across all these methods. It's just that with this sequence-based model, you actually can predict the effect of individual SNPs, but that you can't do with those other methods. And I think that's where the power comes from.
Can I? Sorry. So I had a question. You showed that most of the peaks that you're dealing with, that you are able to predict, are connected to transcription factors. And the other ones, the ones that are not connected to transcription factors, what are they connected to? How you should look for, I don't know, histone modifications or these kind of things? Chromatin states, basically.
Yeah, so I think that's a great question. There's maybe in our first project, there's about 10 to 15 motifs that are not linked to any transcription factors. And oftentimes when we look at them, it's hard. Some of them look like just sequence subcontexts that might be derived from repetitive elements or something like that. But some of them look like motifs that have not been described. But yeah, I guess it's like a matter of working with somebody that can actually do some experiment that's interested in those to figure out what they actually are.
Thank you for the talk. I have one question. Like when you analyzing those motifs, the position weight matrix, I think in traditional methods like Mimi or Homer, I found like for instance with the taxic data, it's easy. It sometimes creates a mess when in a family of TF like ETS or I don't know some or AP1, those family, because those TFs are very similar. I'm curious whether your method can do better distinguish TFs within the same family, like I think also Fox family is another one, because these TFs might have even opposite or distinct functions.
Yeah, so definitely we can recognize different members of the same family, but I think it's always like a manual effort to try to figure out how to put them together to say what actually, like we can describe the motif, but what does it actually represent? What's the name of the transcription factor? That's hard because of this issue of redundancy and many kind of members of the same family looking the same. So yeah, I think there is definitely a challenge there, but we can find different versions or redundant versions of the same motifs.
Yeah, I guess that's the challenge when aggregating information to position matrix. Yeah, thank you.
I'll take the liberty of maybe asking the last question. So I really like your simple idea of empirically assessing the uncertainty in the parameters of the models when you just run it multiple times. I think the results also show that maybe 10 is not enough. How many you need to run and how expensive?
Actually, I thought 10 was it's a really good way to get the general gist of what's going on, because what we see is this dichotomy, like some of the motifs are reproducing 8 or 9 times and some just 1 or 2 times. There's not a lot in the gray area. Yeah, so I guess it's just the cost of running the model 10 times. However, so in our case, it takes maybe 1 or 2 hours on like standard GPUs.